We have been discussing the different role of protein and the nucleic acid molecules in the molecular biology. Now, we will be discussing the different analytical techniques which are used to determine their size, the characteristics, and the properties thereof of these molecules. One important aspect of these techniques is the use of mass spectrometric techniques and the use of different detectors. In this segment, I will discuss the detectors, the role of detector in the mass spectrometric techniques. Now, what happens that when we put a substance in the mass spectrometer, the ion from the mass analyzer, it impinges on the surface of a detector. The detector is there which detects the characteristics of those molecules. And what happens at the surface of detector? When the ion strike, the charge is neutralized either by collection of or donation of electrons. Either electrons, they are collected or they are donated, and the charge is neutralized, whatever the case may be. An electric current, therefore, as a result of that, it flows, and then that electric current is amplified. It is increased and ultimately converted into a signal. An electric current flows that is amplified and ultimately converted into a signal, and that signal is processed by a computer, where it goes into a computer and the signal is processed. Of course, all these ions and that signal, the total ion current, that is the TIC, which is the sum of the current which is carried by all the ions being detected at a given moment. On the detector, on the given moment, all the ions which are detected, and TIC is that. Now, TIC is very useful parameter to measure during online mass spectrophotometry when it is there. And what happens? A plot of ion current versus time complements the ultraviolet trace that is recorded during the chromatography run. That is, it is associated with that. And TIC, the total ion current, is of course independent of the light absorbing properties of the substance. You see, the substance, if it has got a light absorbing property, TIC will be independent of that, and we will get a result which is not due to the light absorption properties of that substance. Now, total ion current, it depends on its ionizability in the instrument. That is, this current, as a matter of fact, is depending upon the ionizability in the instrument, that the substance which we have placed in the instrument, whatever ionization capabilities of that material is, then total ion current is, as a matter of fact, dependent on that.